Welcome back, ladies and gentlemen. So, up to this point, we have sort of been talking about the mechanisms by which cells are capable of controlling and modulating their gene expression. And when we talked about prokaryotic gene expression in terms of those operon systems, we sort of focused on the fact that those cells were, in fact, regulating their gene expression mostly out of a need to be able to utilize resources and utilize energy in the most efficient way possible. And while that's definitely true in eukaryotic cells, there's a whole nother level to why our multicellular eukaryotic organisms are going to address this idea of gene expression. And really, it's going to come down to differential gene expression because of cellular differentiation. So let's back this up a step, right? When we look at the creation of a multicellular eukaryotic organism, we're talking about an egg plus sperm becomes fertilized, and that initial fertilized cell eventually becomes the first starting cell for the offspring. That cell will then go through rounds of cell division. That zygote will go through rounds of cell division to become a blastula, and eventually an embryo and a fetus, and ultimately produce that full grown, quote unquote, baby for that next generation. But then even that baby continues to go through rounds of cell division to go from newborn to infant to toddler, if we're talking humans, to juvenile to eventually an adult. So when we look at all of this, your cells in a multicellular organism are organized. We've got cells that do similar jobs that are organized into tissues. Those tissues are organized by similar job into organs. Those organs that do similar jobs get organized into those organ systems, and it's the collection of those organ systems that ultimately makes up the overall organism. But if we go back to the genetics of the cells in that multicellular organism, every cell in that individual came from that initial fertilized cell, which means the genetics don't change. If I look at your body and I take a cell out of your brain, a cell out of your skin, a cell out of your intestines, all of those cells contain 46 chromosomes. All of the same genetic information exists in every cell type in your body. So it comes down to the orchestration of differential gene expression that determines what type of cell it is and the job that it performs, because it's not going to access all 46 chromosomes worth of information to do every job in your body. It differentiates. It's just like you going off to college or to a trade school or getting on the job training, right? Not every person in society does the same job, and as a result, although they may have the capacity to do them all, you get trained only on the job you're doing. Hopefully that analogy kind of makes sense. So when we look at the transformation from zygote, fertilized egg, to adult, it results in that gene division, or that cell division, I should say, itself, the cellular differentiation, as well as this idea of just morphogenesis. So morphing, giving you structure, giving you shape that gets created over those rounds of division and gets created based on the set of genetic instructions that that particular cell or that particular subset of cells ends up following. So let's just make sure we've got some terminology down here, right? So cellular differentiation is the process by which cells become specialized both in their structure and in their function. And that's going to come as a result of what subset of genetic information does that cell actually access and express. The morphogenesis is the physical process by which the organism gets its shape, but that is still very much genetically controlled. So differential gene expression results from genes being regulated in different types of cells. If we look at even just that initial zygote, that zygote will go through rounds of division to become a blastocyst. That blastocyst then will end up becoming a gastrula, which starts producing things like these mesoderm layers, which will become things like cardiac muscle, skeletal muscle. The endoderm layer, all right, or internal layer, things like lung cells and thyroid cells. Um, and then you get your uh, ectoderm, so cells of your skin, your neurons, pigment cells, and then even this sort of additional stuff out here. We've got to take that initial ball of cells and produce not only the baby, but also the placenta in the mother. So that's what you're looking at here in this layer. All right. Germ cells, eggs or sperm, depending on whether it's male or female. So that initial round of cells starts dividing and starts taking on different components very, very early on. And the determination or is the commitment to a cell to its final fate. And that determination precedes the actual differentiation, but it does come down to turning on and turning off certain genes and certain layers of cells to ultimately be able to determine the fate of that cell type. So how can this occur? Well, there's a large number of mechanisms. We'll talk about some of them. One of them are these things called cytoplasmic determinants, which is pretty cool. So let's go back to the fact that that initial zygote has to split not only into baby, but also placenta. So if we use that as an example for this, we look at that unfertilized egg. Here's your nucleus, which contains half the genetic information. Dad's sperm's going to come and bring the other half. But inside that unfertilized egg, there are already these molecules, these cytoplasmic determinants that are produced by the mother in the creation of that unfertilized egg, where you can see the high concentration of this particular molecule here and this particular molecule over here. So that when we get fertilized and that spurs the initial rounds of mitotic division, you can see that most of those orange cytoplasmic determinants end up over here. Most of these green ones end up over here. So if we assume that this orange one is going to force or influence the gene expression in this cell to become placenta, where the green ones over here are going to influence this cell to start forming baby, that's a great example of how these cytoplasmic determinants influence that early development. So that's just a really cut and dry one. Placenta goes one way, embryo goes another, but they are all going to be initially interconnected until baby ultimately becomes born. You cut the umbilical cord and they are now two separate entities for sure. We can look at those cytoplasmic determinants and there's a whole number of things that exist in terms of starting to build that brain structure. That is one of the very early organ systems that gets built. The skin being another, right? And all of these have to do with the way that that cell division occurs, isolating or concentrating certain cytoplasmic determinants in one cell line as opposed to another. Another important source of developmental information actually comes from the environment, so surrounding cells, especially in nearby embryonic cells. And that's this idea of induction. So induction is just the process in which signal molecules from one set of cells influences the transcription in a targeting cell, a nearby cell. And those interactions between the signal molecules from one set of cells to the influence that they have over neighboring cells can also induce specialization in different cell types. So if I am a cell over here that based on my set of cytoplasmic determinants perhaps, or just general differentiation in my gene expression has determined that I am going to become part of the heart, then they may produce signaling molecules, inducers, that then go influence neighboring cells to tell them to be other layers of the heart. And they do that through these signal transduction pathways. Basically, they're just signal relaying pathways that then ultimately influence the gene expression, the transcription, the translation that ends up happening in that nucleus nearby. Sort of think of it like peer pressure, right? So you have a group of friends, somebody in that friend group has decided that they are going to run for student council and they sort of influence everybody else around them to sort of join them in running as well, right? So we're going to make all of student council be my friend group, kind of something like that, right? So it's sort of that peer pressure. Or somebody in a group decides that they are going to go to school to be a nurse and sort of spirals into that whole friend group deciding, oh yeah, that sounds like a good idea. I think I'll go do that too. Um, and so it's sort of collective group thought. One set in that group already is predetermined and starts influencing the cells around it.
Morning, everybody. Welcome to another edition of the Lab 207 webcast. My name is Mr. Kite, and I'll be hanging out with you today. We are about to wrap up our series on DNA. Final topic to be discussed is cell differentiation, or differential gene expression. Essentially, two sides of the same coin. So let me get you your objectives, and then we'll start talking about some things. By the end of the video, here's the stuff that I need you to know. First, describe cellular processes that lead to cell differentiation. Second, understand the importance of homeotic genes in the establishment of body plan. And finally, explain the importance of the p53 gene. So here's what we got. I'm going to tell you before we even get going that this topic can be covered in excruciating depth if you are in molecular biology or something like that. We're just going to go over an overview, which is probably about all that you're going to need for now. So stick with me as we walk through some of this stuff. First thing I talked about is some helpful vocabulary. There's some vocabulary and some words that are used differently in this lesson than would normally be used in other lessons, so I just kind of want to get you a baseline for what the words are. First one is differentiation. All right, for today's topic, when we are talking about differentiation, we are talking about cells that are alike becoming different from one another. So if you've got two cells in your initial zygote that are the same, one of them might become a heart cell, one of them might become a brain cell. That is differentiation. Morphogenesis is the process of actually developing a form. So in differentiation, the cells would say, hey, I'm going to be a heart, or I'm going to be a brain. Morphogenesis is where the cell actually becomes a heart or becomes a brain. Cytoplasmic determinants are signals in the cytoplasm that help to determine what cell is going to be what. We'll talk about that in more detail in a second. Induction is one cell influencing another cell and helping it to decide what it is going to become, kind of like peer pressure. And finally, determination is kind of like at the end of the line. Once a cell has been determined, then it is set to become whatever cell it has been determined to become. So let's go ahead and talk about some of these things in a little more depth. If my slide will click over. Excuse me. All right, so zygote to embryo, biological organization. So the whole reason that we are talking about this idea of cell differentiation is there's kind of an interesting thing to consider. Every one of your cells in your body has the exact same set of DNA in it, except for eggs or sperm. We've talked about that. But all the somatic cells in your body, they've got the exact same DNA, and yet they are all different from each other. I mean, skin cells are generally like skin cells, but skin cells aren't like muscle cells or bone cells or anything like that. So it's interesting to think that any one of your cells has the instructions to be any other type of cell, yet it is particular that it is a skin cell or a muscle cell or whatever else. So that is where we get differential gene expression, which is different genes being expressed causing different cells to do different things. Need to recognize this biological organization hierarchy. So you've probably heard this before, but I want you to remember that cells are organized into tissues, tissues are organized into organs, organs are organized into organ systems, and organ systems make up an organism. And the interesting thing is you've got like one type of cell or many types of cell, depending on how you look at it. At the level of a tissue, you've got kind of one type of cell, but if you're looking at an organ system, you got all kinds of different cells working together. So I wanted to get that organization hierarchy in your head before we moved forward. So here's differentiation and morphogenesis. And this is like, all right, so we're going to start out with our initial zygote. For the first several divisions, all the cells in the zygote are exactly the same. So it starts out as just one little zygote, and then that thing starts dividing in half and dividing in half and dividing in half and dividing in half. And it makes many divisions. I'm sure this number is going to be wrong, but I think it gets up beyond 64 cells or something like that before the cells actually start to become different from each other. They are all alike initially. As that process of division happens, though, we start to get the process of differentiation and morphogenesis. Differentiation is where a cell is kind of told what it's going to become. Morphogenesis is where it actually starts to become that thing. Morpho is form, genesis means to become, so it's becoming its form. One of the ways that a cell is told what it's going to become is through cytoplasmic determinants. And this drawing here is really interesting. It's a drawing from the 50s, and it kind of talks about how this works. So right here would be our fertilized zygote. All right, so we've got the egg. Obviously, what you're seeing right here is the egg. What you don't see is that a sperm has come and fertilized this egg. So it's got a full set of genetic information. It hasn't started to divide yet. But the thing that's being highlighted here is within the cytoplasm of the egg, the cytoplasm is not the same. And throughout this cytoplasm in the egg, some sections have got like more mRNAs or different types of mRNAs. Some have got other cytoplasmic chemicals. Just recognize that like this section right here might have a high concentration of one specific type of mRNA. As our zygote goes through and starts to divide, this chunk of cytoplasm that we originally talked about is going to get moved around as the cell goes through a couple divisions. So the first division would be right here, and then right here you can see that there's two cells. Here's one, here's the other one back here. This time it's going to divide this way. And you can see, following this yellow, that as the zygote divides and becomes a blastula, that initial chunk of cytoplasm that had a high concentration of whatever mRNA or chemical or whatever is staying with one section of cells. That mRNA is going to signal the DNA within these cells to start turning on or turning off genes, which is going to cause those cells to become specific types of cells. So that is a cytoplasmic determinant, and it is a signal within the zygote that is telling the cells which genes to turn on and which genes to turn off. Another strategy that cells use to signal what they are to become is called induction. And this is kind of like peer pressure from other cells. So we just talked about cytoplasmic determinants inside the cell telling it what it's going to become. Once those cells start to become differentiated into what they're going to be, they can exert signals to the cells that are next to them. This might look like signaling each other through cell membrane receptors. It might look like sending out growth factors. Either of those things could cause the cells next to each other to start to become the same type of cells. So you could have, let's say right here you have got a brain cell. Right here, undifferentiated, undifferentiated. This one might continue down the path to a skin cell, but this one could send some signals over towards that one, causing it to also become a brain cell. So that would be induction.
So cellular differentiation is definitely marked by production of tissue-specific proteins. So when we look at different cell types, there are sort of these master regulatory genes that then influence the production of tissue-specific proteins. So something like these myoblasts, right? So these are protein-specific, or I should say muscle-specific proteins are being produced in these cells. So we've got this embryonic precursor cell, right? This is sort of a blank stem cell. It has those master regulatory genes to be a muscle cell, to be a neural cell, to be a skin cell, and then those other specific genes that go along with those tissues, but that master regulatory gene is shut off. Okay? In fact, probably all of the tissue-specific master genes are basically turned off in that embryonic precursor. But as that cell divides, that stem cell divides and starts to become determined, it now turns into a myoblast, so a muscle-specific stem cell. That myoblast now has become determined because that regulatory gene for that cell has determined it's going to be a muscle cell, and that master regulatory muscle gene has been turned on. But at this point, we're not technically a muscle cell yet, so we're not producing those muscle specific genes quite yet. So I am a determined stem cell, sort of what we would talk about as an embryonic stem cell as opposed to an adult stem cell, where an adult stem cell is already down the path of being tissue specific. Um, but it may be able to be a couple of different types of cells in that tissue. That part hasn't been determined yet. Once that myoblast becomes part of an actual muscle cell fiber, it's fully determined, it's an actual functional cell, will now utilize that master regulatory gene, go through transcription and translation. But what that ends up doing is producing transcription factors for those other muscle specific genes down the line until we actually produce things like myosin or actin or other sort of proteins that are specific to being that actual muscle cell itself.